a novel approach to drug discovery using latent space manipulation of a variational autoencoder in tandem with a graph neural network and document simulation. Hypothesis: if a variational autoencoder uses latent space manipulation across multiple iterations, then it will produce drugs with higher simulated binding affinity. So, recent advances in drug discovery have significantly enabled us to use deep learning models for molecular representation, generation, and screening. Graph neural networks have emerged due to the ability to use nodes and edges to represent these atoms and bonds. This allows them to achieve remarkable molecular representation and has also allowed them to extrapolate molecular properties, allowing them to make predictions of how multiple molecules will interact with each other, and also allows them to predict new properties of those molecules. Another thing is the VAE, which is a form of generative model that forms a latent space after being trained on molecules. One aspect of the latent space is that we're able to manipulate it to specify what molecules the model should generate. Another thing is physics-based molecular docking is a widely used method that estimates how well a ligand and protein bind together, resulting in a binding affinity score. This is just a representation of the latent space given by Geeks for Geek, and this is pretty much how the VAE works. It's trained on multiple molecules to create this 2D vector, sometimes even 3D or 4D spaces. And then when we want to create a new molecule, you just take one point from here, run it through a function, and you create an entirely new molecule. So we constructed the database using Tremble and the Tremble API. We then use Lipinski's rule of 5 to narrow down the number of drugs we should use. Then we use the automated, the main thing about our project is the automated pipeline. And this is pretty much where the VAE will generate a molecule, will generate actually 1,000 molecules. The GNN will predict those 1,000 molecules binding affinity scores and only take the top 100. Those top 100 will go through the physics-based docking system, which will then give us the real binding affinity scores. And then we can use all this information to help update the latent space of the VAE to create brand new molecules that are better each time. So we set up the machine learning environment by using Google Colab. We made sure to use Python and install dependencies like NumPy, Pandas, and Torch. Then we created the model using mostly internet resources, past research papers, and GitHub repositories. And the Docker simulation was we mainly used Autodog Vina, and we pretty much used the Tremble API to run it. So these are the results of the VAE. Essentially, you have five main metrics that we looked at over the course of 100 epochs, which is the reconstructed accuracy, validity, uniqueness, novelty, and diversity. This data is essentially telling us that the model was able to create was able to create a lot of good drugs and made them very unique and different from each other. However, because we're zooming in so much into our latent space, the diversity and validity of them did reduce over time. This is the result of the GNN model over 300 epochs, and this is a very good results. It's showing that the RMSE, which is the root mean square error, has reduced over time. Although validation R squared, which is the correlation between the molecules and the binding score, did improve. These two scores alone shows that the GNN was very efficient at predicting binding affinity scores. So this is the final result of our pipeline. We can see over many iterations that the scores improved over time, and these scores are the qualified best binding affinity scores from each iteration. You can see the first iteration, after, sorry, 5 iterations, we had negative 3.7. After 50, we had negative 7.7. And this shows a very strong correlation. Although it looks backwards that the numbers are negative and that seems to decrease, the negative 7.5 is stronger than negative 7 because of how binding affinity is scored. And the strong correlation coefficient and low p-value show that there's a very strong correlation between the number of iterations we do and how well the drug comes out. On top of this, the slope of negative 0.083 shows that after one iteration, we can estimate to get an improvement of 0.083 in the binding affinity score. However, this score does start to plateau after 100 epochs due to model convergence. And so the main application of this is that drug discovery is very costly and has a very high failure rate. However, by using an automated environment like this, we can allow researchers to prioritize the most promising compounds for synthesis. Furthermore, the architecture is designed to be very flexible, allowing us to create brand new drugs for different proteins just by trading out the data sets. So finally, as you can see, there's a very clear correlation between the number of iterations we do and the binding affinity. One simple thing we can improve is the VAE. We can do this by improving the number of molecules we have, the molecular properties we give it, and potentially using a hybrid model. This can all go to improving the amount of drugs we make and how well these drugs can fit into our binding affinity scores.